Chromosome mutation. Chromosome mutation. A mutation that occurs as a result of errors in crossing over during meiosis as well as of certain mutagens. It affects large segments of DNA containing many genes. Crossing over is the process of exchanging segments of chromosomes between homologous chromosomes and produces recombination. Types of mutation. Point mutation. Changes in one or a few nucleotides. It occurs at a single point in the DNA sequence. Gene mutation. Changes in a single gene. Chromosomal mutation. Changes in a whole chromosome. Types of changes in chromosome structure. Point. A change in one base in the DNA sequence. Substitution. One or more bases in the sequence is replaced by the same number of bases. Inversion. A segment of a chromosome is reversed end to end. Deletion. A base is deleted from the sequence. Insertion. A base is added to the sequence. Duplication. Part of a chromosome is abnormally copied, duplicated. Translocation. A piece of one chromosome breaks off and attaches to another chromosome. Examples of diseases linked to the mutation. Substitution, e.g. sickle cell anemia. Inversion, e.g. Opitz-Kveja syndrome. Deletion, e.g. cystic fibrosis, Cre-du-Chat syndrome. Insertion, e.g. one form of beta thalassemia. Duplication, e.g. some cancers. Translocation, e.g. Down syndrome, chronic myeloid leukemia. Mutagens can cause mutations in DNA and raises their frequency above natural background levels. May directly damage DNA due to their actions. May be carcinogens because numerous mutations can lead to cancer. Types of mutagens. Physical mutagen radiation. Ionizing radiations, e.g. X-rays, gamma rays. Alpha rays, beta rays, electrons, neutrons, protons, other fast-moving particles, non-ionizing radiations, e.g. ultraviolet, visible light. Temperature, the thermal stability of DNA and the rate at which other substances react with it are both impacted by temperature. Chemical mutagen. Base analogues, intercalating agents, metal ion alkylating agents, biological mutagen. Transposons and insertion sequences, viruses, bacteria. Results of mutations. The position of a gene on a chromosome can affect its expression. Moving genes from one location to another can affect their expression, especially during early developmental stages. In humans, significant changes to the position of many genes can prevent proper fetal development. Lethal deletion can cause significant loss of genetic material, most likely to be lethal. Lethal, deleterious, neutral, beneficial. They are much more likely to be deleterious or lethal. Mutations, DNA repair, and evolution. Mutation occurs during DNA replication. Serves to place organisms at risk for disease, on the other hand, serves to diversify the overall population, leading to evolution. DNA repair. In most cases, the extensive network of DNA repair machinery that exists in the cell halts cell division before an incorrectly placed nucleotide is set in place, and a mismatch is made in the complementary strand. Hereditary mutation. If the repair machinery does not catch the mistake and the mutation is established in the cell, then the mutation can be inherited in daughter cells or in embryos, if the mutation has occurred in the germline. Mutation and polymorphism. Mutation. Any alteration in the DNA sequence. Biologists use the term single nucleotide polymorphism, SNP, to refer to a single base pair alteration that is common in The population. Polymorphism. The occurrence of two or more different forms, morphs, in the population of a species. Any genetic location at which at least two different sequences are found, with each sequence present in at least 1%, somewhat arbitrary, of the population. It is a normal variation or one that does not directly cause disease. If the frequency is lower than 1%, the allele is typically regarded as a mutation. Cf. Unequal crossing over.